In chapter 4, we're going to take a tour of the cell of animals and of plants and learn all the different organelles that are within those cells and the importance of those cells and actions and functions that those cells and organelles have. In lab, you're also going to cover quite a bit of the cell. So this is a little bit um, of review for both the lab and help for the lecture. So all organisms are made up of cells. We have tons of cells that make us up. Cells turn into tissues, groups of tissues turn into organs. Cell is the simplest collection of matter that can be alive. So there are actually unicellular, one-celled animals that are living. They're related by their descent from earlier cells. So in order to grow a liver cell or a liver in a petri dish, you have to take from similar cells or from another liver. Those cells can differ substantially from one another. They do share common features. So most cells are too small to be seen by the unaided eye, and as a result, we'll use microscopes in order to see specific characteristics of those. Light microscopes are the microscopes that you're probably the most familiar with. That's where light is passed through a specimen and then through a glass lens, and there should be a lab that you'll do related to the microscope and understanding how it refracts or bends light in order to magnify the objects for you. So um, the three important parameters of microscopy, just to keep in mind: magnification, so they increase the image's size, right? Resolution is the measure of clarity of an image, and contrast are visible differences in parts of the sample. The light microscopes basically magnify about 1,000 times the size of the actual specimen. So at some point, you can't really see much. If you stuck your finger underneath the microscope and multiplied it by 1,000 times, at some point, you're going to have to increase your clarity by focusing better so that you can see specific parts of your finger. But your finger isn't in the view going to look 1,000 times bigger necessarily. Various techniques enhance contrast and enable cell components to be stained or labeled. So we're able to, on certain cells that are very light or very translucent, stain them with specific stains so that certain components pop out to you so you can study and see different things. Most subcellular structures, including organelles, are too small to be resolved by light microscopy. And so as a result, you have to use different types of microscopes that we'll talk about in a second. So these are a bunch of cells. There's, I guess, about 4 or so that you can see in the images, top and bottom. And then on the right-hand side, top, there's about 5 that you can see. The only organelle that we are really able to see and spot in these is the nucleus, because the nucleus is rather large and it's also very opaque. So this is some technology in which you're able to see different fluorescence. And if you notice, 50 micrometers, 10 micrometers, 50 micrometers, so different sizes of objects that we're seeing. Basically, with our naked eye, we're able to see pretty small things. We're able to see things that are in the micrometers, which are tinier than a millimeter. A centimeter is usually one little dash on your ruler. So we're able to see certain objects, but we use the light microscope once we get about to 1 millimeter or so to be able to see things a lot closer and a lot better. Keep going down, light microscope kind of peters out about the smallest bacteria. And then when we get into viruses and the things below that, ribosomes, proteins, and lipids, those are components of the cell and organelles. And so in this case, we have to utilize the EM microscope, electron microscope. Electron microscopes are used to study subcellular structure. So light microscope looks at the whole cell, electron microscope looks at parts of the cell. And then there's scanning electron microscopes that there's 2 different types of electron microscopes, scanning and transmission. So scanning focuses a beam of electrons onto the surface of a specimen, and it makes the images look 3-dimensional, whereas a transmission focuses the beam through, transmits through, scanning just on top. And then transmission is mainly used to study the internal structure of cells because we get to see what's inside. So this is what you would see with a scanning microscope. Cilia are like little fingers or hairs that help with movement of the cell. And the image to the left just kind of shows you it's a pretty big machine to see an electron microscope. Transmission electron microscope, same thing, pretty big machine. But now we're actually able to take those cilia, those little fingers, and cut them in half and see what it looks like on the inside, because it's transmitting through the what it's looking at. Cell fractionation breaks up cells and separates the components using centrifuges. So, you know, a centrifuge is something that spins super duper duper fast, and so it's breaking things apart. Heavy stuff will fall and light stuff will go to the top. But that enables scientists to figure out the functions of organelles. And then not really all that important, but biochemistry and cytology are two subunits of biology that help correlate cell function and structure. So now we're going to look at eukaryotic cells specifically. These guys have internal membranes that compartmentalize their functions. So there's certain, it's like having walls in your home that separate different rooms so that you're not eating maybe in the same place that the restroom is, right? Or you're not showering where you're, I don't know, where you're watching TV. There are walls that compartmentalize and close things up to separate them. The basic structure and function unit of every organism is one of two types of cells. You're either a prokaryotic cell or you're a eukaryotic cell. If you remember from chapter 1, I believe, we talked about prokaryotic cells as being things that would be bacteria, very, very simple, very primitive type animals. And eukaryotic cells are more complex. So prokaryotic cells are domains bacteria and archaea, and then eukaryotic cells are things that by and large are more advanced: protists, fungus, animals, and plants. The basic features that all cells will have: they have a plasma membrane, they have a semi-fluid substance called a cytosol, they have chromosomes, and they have ribosomes. The plasma membrane is what encases the entire cell so that the guts don't spill out. The cytosol is sometimes called the matrix or the intercellular matrix. It's the stuff inside the cell so that it doesn't lay flat, so that cells are kind of like a water balloon, or there's jello inside of a balloon. It's like a semi-fluid substance that keeps things cushioned, all the organelles cushioned. Kind of like blood is our matrix, blood and fat keep our organs cushioned inside of our body so that through gravity they don't all just fall down through our legs to our feet. Chromosomes are where genetics are found, right? That's our genes. And ribosomes are organelles that make proteins. In eukaryotic cells, most of the DNA is in the nucleus, and the nucleus is bound by a double membrane. Prokaryotic cells, though, don't have a nucleus. They just have DNA that's just kind of squiggled around. It's like a ball of spaghetti. And they don't have any membrane-bound organelles. Both prokaryotic and eukaryotic cells will have the cytoplasm, and around it also the plasma membrane. So that's a really good test question or quiz question. What are two components of cells that both eukaryotic and prokaryotic cells have? Cytoplasm and plasma membrane, and DNA would be another one. And then know the differences between eukaryotic and prokaryotic cells as well. You can see here, this is a prokaryotic cell, it's a bacteria. You have ribosomes in here and you have a nucleoid region. That's that spaghetti type stuff. It's not bound up by a membrane at all, but that kind of floats around in the cytosol. And what surrounds the cytosol and everything else is the plasma membrane. Eukaryotic cells are usually bigger than prokaryotic cells. So, and by, as many as 20 times. So they can be 1 to 5 micrometers, but eukaryotic cells are 10 to 100. The exact number doesn't matter. I would just understand that eukaryotic cells are bigger than prokaryotic cells. The plasma membrane is a selective barrier that allows sufficient passage of oxygen, nutrients, and waste to service the volume of every cell. So the plasma membrane holds the cytosol and all the organelles inside, but it also allows, it's not like completely like a rubber glove where nothing can go inside of it. It's semi-permeable. So it would be like a rubber glove versus a gardening glove. If you've ever gardened in the mud with the gardening glove, you still get mud underneath your fingernails and on your hands, right? It's semi-permeable. It allows some things to come inside. The general structure of a biological membrane is a double layer of phospholipids, and we're going to cover this ad nauseum when we get into the chapter of plasma membranes and lipid bilayers and all that kind of stuff. You're going to understand that quite a bit better. But basically what it means is that a bi means two, so it's made up of two components. And you have a head region, which is hydrophilic. Hydro means water, philic means to love. This part loves water. So the hydrophilic region likes to touch the outside or the inside of the cell where there's a lot of water. The hydrophobic region are the tails. And the tails, hydro means water and phobic means afraid of. So these guys don't want to touch the tails. And as a result, or the tails don't want to touch the water. So they want to be protected. So as a result, it makes this bilayer in which the hydrophilic heads are on the two outer layers and the hydrophilic tails are placed in the middle. And a lot of times you'll also have lipids, which are fats, or proteins that are embedded within that plasma membrane that also facilitates transports of things. So you can think of them as like a straw or a slide to allow things to move across the membrane into and out of the cell. One thing to know is that although eukaryotic cells are larger than prokaryotic cells, they're still very, very tiny, and they're limited in size. And there's a method to the madness. So by having little bitty teeny tiny cells, you actually increase the surface area of these cells, the ratio. If the bigger the cell is, the more guts it has versus outside area. The smaller it is, the larger the surface area. So small cells have a greater surface area relative to their volume, and a lot of work can happen when you have a greater surface area, when you have more plasma membrane to deal with. And so that makes it really important. So if you look here, let's say it's a 1-inch block. One little tiny 1-inch block has a surface to volume ratio of 6. If you were to increase that block 5 inches, so it's 5 by 5 by 5 cube, you only have a surface volume of 1.2. But if you have the same volume that you're filling with bunches of little 1 by 1 squares, little 1 by 1 cubes, you have the same amount of surface area. So it's much, much more important for cells to stay smaller and just to multiply than to grow larger. So we're not made up of just one gigantic cell or 3 gigantic cells. We're made up of tons and tons of smaller cells. So when you start out egg and sperm fuse, it will grow a little bit, and then it will start to replicate and make more of itself, as opposed to just getting bigger and bigger. A eukaryotic cell has internal membranes that divide the cell into compartments. Those are called organelles. So just as we have organs inside of our body that make our body do the work that it does, what makes the cell do its work is organelles. And our liver does something different from our heart does something different from our brain. All organelles do something different from one another as well. So let's watch this video.
Even if you're just studying, a lot of work is going on in your cells. Let's zoom down and check it out. We've arrived in the space between two cells. A sticky coat called the extracellular matrix holds the cells together. Each cell is surrounded by a flexible plasma membrane with an incredible number of projections, docking stations, and channels. Let's dive into one of these channels to enter a cell. Whoa, look at this place. These girders and cables make up the cytoskeleton, the structural framework of the cell. They also serve as tracks for transporting cargo from one place to another. All this activity in the cell requires energy, in the form of ATP molecules, which are made here in the mitochondrion. Notice the outer membrane and the inner membrane with its numerous infoldings. Many of the molecules involved in making ATP are built into the inner membrane. All those folds increase the inner surface area, enabling more ATP to be made. Moving towards the nucleus, we pass by layers of internal membranes. The nucleus is enclosed by a double membrane called the nuclear envelope. Let's enter the nucleus through a pore. The nucleus houses the genetic material of the cell, DNA, which carries the blueprints for making the cell's proteins. Almost 2 meters of DNA is crammed inside the nucleus. How does it all fit? The DNA is wrapped around proteins like thread wrapped around spools. Look, this section of the DNA has unwound, and a different protein has attached to the DNA. DNA is being used as a template to make mRNA. mRNA molecules travel from the nucleus to the cytoplasm, carrying the instructions for making specific proteins. In the cytoplasm, a ribosome clamps onto a strand of mRNA. The ribosome ratchets along the mRNA, building a new protein. Some proteins stay in the cytoplasm. Others, like this one, are processed in special compartments within the cell. Protein processing and certain other metabolic activities occur in the endomembrane system, the cell's network of internal membranes. The endoplasmic reticulum, or ER, is part of the endomembrane system. There are 2 types of ER, rough and smooth. Rough ER is covered with ribosomes. Smooth ER lacks ribosomes. Lipids are made in the smooth ER. Let's go inside the rough ER. Note that you can still see the ribosome on the outside surface. The ribosome is manufacturing a new protein, which continues to grow inside the ER. Completed proteins move to the edge of the rough ER and depart in a vesicle that buds off from the ER membrane. Some vesicles fuse with the Golgi apparatus, another component of the endomembrane system. In the Golgi, proteins undergo further processing. Finished proteins are then packaged in vesicles that pinch off from the Golgi and are transported along cytoskeleton tracks. Some vesicles bind with the plasma membrane, secreting their contents outside the cell. Other vesicles, called lysosomes, contain digestive enzymes. Here, a lysosome fuses with a worn-out mitochondrion and breaks it down. Each of the trillions of cells in your body is a dynamo of activity, requiring millions of ATPs every minute. But most people are unaware of all this activity in their cells.
So that's an overview of everything that we're going to look at right now. This is an image. It's in your book. It's a really fantastic image showing you exactly what a cell looks like in all the parts of the cell. So as you quiz yourself, use this image to point at things and come up with the function of each of these organelles. This picture is an animal cell. We'll get to talking about how I can tell it's an animal cell versus a plant cell. One reason is because animal cells only have a plasma membrane around them, and so they're more round. And then when I look at this cell, it's more rigid. That's a plant cell. Plant cells have to be rigid because they don't have muscles like we do to hold them up. Instead, they have to have a hard cell so that they're able to grow big and tall instead of being kind of limp and wilting over all the time. I can also see that they have an organelle that the animal cell did not have, which is a chloroplast, which is how they do photosynthesis different than animals do. So first, let's talk about DNA and what's inside that cell that carries the genetic information. So genetic information, or DNA, is housed in the nucleus and carried out by the ribosomes. So ribosomes use the information that DNA has to make proteins. DNA is our genetic information and material. Ribosomes will go to the nucleus get the paperwork from the DNA and then go take it to the manufacturer to make a protein. Nucleus contains most of the cell's genes. It's usually the most conspicuous organelle. We saw that already. The nuclear envelope is what encloses the nucleus and separates it from the cytoplasm. And it has a double membrane as well, just like the plasma membrane has. So it has two membranes. Plasma membrane is just one, but each of those two membranes has a lipid bilayer. So I would just know nucleus has a nuclear envelope and inner and outer membrane. There's little pores on it which allow things to come in and out. And there you see it using a transmission microscope. Kind of looks like the moon, has craters of the moon. So in the nucleus, DNA is organized into discrete units called chromosomes. So your chromosomes are the units of DNA. Each chromosome is one long DNA molecule with its associated proteins. DNA and proteins of chromosomes together are called chromatin. So chromatin is DNA plus proteins. Chromosomes is just the DNA. Chromatin condenses to form discrete chromosomes as a cell gets ready to divide. So most of the time, unless the cell is preparing to divide, and we'll talk about this in mitosis and meiosis, it's just a bunch of squiggly mess. Then it starts to condense and makes that little kind of X that you and I recognize as DNA. That I think I guess it was in Jurassic Park was like the little, was it DNA, the little cartoon or something in the beginning of the story. The nucleolus is located within the nucleus and is the site of RNA synthesis. So the nucleus has its own nucleus, and we call that the nucleolus. Ribosomes are complexes of ribosomal RNA in a protein. Basically, the important thing to know is that ribosomes carry out protein synthesis. Synthesize means to make, and so ribosomes make proteins, or they pass along the information to get those proteins made. So that happens in two places. It happens in the cytosol, or it happens outside the endoplasmic reticulum, or the nuclear envelope. So, and I'll show you what these things both look like. And we know them as bound ribosomes and free ribosomes. And when we look at the endoplasmic reticulum, you're going to see those ribosomes on there. So I don't see them here. There is, within the cell, we have this thing called an endomembrane system. So it would kind of be like our lymphatic system, and it would include all the organs that make that up, or our circulatory system and all the organs that make that up. So this endomembrane system is made up of 6 different things. The nuclear envelope, which is what covers the nucleus. The endoplasmic reticulum, we haven't really talked about yet. Golgi apparatus, which we have not talked about. Lysosomes, vacuoles, neither of which we've talked about, and that plasma membrane. These components are either continuous or connected through transfer by vesicles. Vesicles are vehicles that might send information in between these organelles. And when it says that they're continuous, meaning that they touch one another. The endoplasmic reticulum accounts for more than half the total membrane in many eukaryotic cells. So the endoplasmic reticulum is it's smushed up really tight, but if you were to pull it apart, it's kind of like an accordion. It's folded and curled and folded up, but it would stretch out really long. So it has a super, super long and very, very big membrane. It is continuous with the nuclear envelope, meaning that it touches it, it's connected to it. And we've got smooth ER and rough ER. Smooth ER doesn't have any ribosomes on it, and rough ER is where the ribosomes are bound to it. So this is our endoplasmic reticulum, and you can see that the purple is the nuclear envelope. It is connected to the nucleus in that way. And you have smooth parts and you have rough parts. What I see here is all basically rough because I see a lot of ribosomes connected to it, but maybe right here in this little section that looks pretty smooth. So this video just kind of shows you, I guess they've kind of got some sort of a light that is showing um, how big that endoplasmic reticulum is. This is just a really bad video that shows you, let me see if I can, oops, that just kind of shows you what that looks like. It's so bad it's not even worth looking at. So I've said that the endoplasmic reticulum has two parts to it, and each part, smooth or rough, each part does something different. So the smooth ER synthesizes or makes lipids. Lipids are fats. So I can remember that because smooth ER is your skin is smoother if it's full of collagen and fat, right? Babies have smoother skin than older people do who've lost that. It metabolizes carbs, it detoxifies drugs and poisons, and it stores calcium ions. So understand and know that, so that if you were given a scenario, you would be able to tell me which organelles would be responsible for something. So if I said somebody has overdosed on drugs, which organelles within the cells would help that out? Or another good question is, since we know that it detoxifies drugs and poisons, would we have a large or a small amount of smooth ER associated with our liver, knowing that our liver detoxifies things? Well, we'd have all of our liver cells probably have a whole lot more smooth ER than other organelles. The rough ER, since it has ribosomes, it will secrete these little proteins, glycoproteins, which are proteins and carbs. It distributes transport vesicles. Transport vesicles are just little vehicles, little balls surrounded by a membrane that move around and do stuff. And it is a membrane factory for the cell, basically. So the rough ER is the warehouse. It's a manufacturing plant for the cell. The Golgi actually is more of the warehouse, I suppose, shipping and receiving center. So it'd be like the place where all the packages go to UPS before they get shipped out to all the homes. The Golgi is a flattened membranous sac. It's called cisternae. The functions of the Golgi are that it modifies products for the ER, for the endoplasmic reticulum, so it kind of fixes things for them. It manufactures certain macromolecules, and then it sorts and packages things into transport vesicles. It is the UPS of the cell. So here's our Golgi. If you remember, this was our nucleus right here. This was our ER, endoplasmic reticulum. And then our Golgi is separate, and it's kind of smoother and globular and kind of folded up also. Here you can see these little vesicles that are kind of blobbing off and taking information and things to or away from the Golgi. Here's some, with the electron microscope, some of that movement. Things being moved into the Golgi. You can kind of see some things getting moved out. All these little, mainly I see vesicles being moved in, and just every now and then a few that are getting spit out like little shooting stars. So really neat. Now the lysosome, also part of that endomembrane system, is basically the digestive component of our cell. It's the trash can. It's the trash compactor. It's made up of lots of enzymes that are pretty acidic, and it will engulf things and eat them up, dissolve them up. The lysosome is kind of interesting because it has to have a special membrane, otherwise it would dissolve itself, right? But it doesn't. It's unable to. It doesn't do that because of its membrane. So sometimes cells can engulf another cell by something called phagocytosis, and this forms a food vacuole. And so lysosomes food will fuse with that food vacuole and start to digest it. Sometimes they will use their enzymes to recycle the cell's own organelles and micromolecules. That's called autophagy. Phagy is to eat, and autophagy meaning, I guess, eat itself. But basically, it's just cleaning things up. It will clean things up that aren't supposed to be there. There's too much of that are dying, that kind of thing.
Like secretory proteins and some other proteins, proteins destined for lysosomes are made on ribosomes bound to the rough ER and move through the endomembrane system. In this case, the lysosomal protein-containing vesicle that buds from the Golgi apparatus is the lysosome itself.
Okay, and then this image tries to show you how that lysosome, these are food vacuoles, so that engulfed food. It's like the mouth, right? Phagocytosis, these little vacuoles get moving. Lysosome goes bloop, and it attaches to it, and then it dissolves that, digests that food. And then this is just kind of neat. It kind of shows the actual process of phagocytosis there. Vacuoles are basically storage devices. Vacuoles are big vesicles, way bigger than a lysosome, and they're derived from the endoplasmic reticulum and the Golgi. So inside the vacuole, what is in there is going to differ from what's outside the cytosol. And we have different types of vacuoles. There's food vacuoles, which are formed by phagocytosis. There's contractile vacuoles, which are usually in things that live in water. And so what they'll do is they'll pump extra water out of the cells since they're so saturated with water wherever they are. There's central vacuoles, which are found in plants. And what they do is they're a big compartment that can really stretch out when water is abundant, and then they let go of that water when water isn't all that abundant to make sure that it gets water to the plant. Certain vacuoles in plants and fungi carry out enzymatic hydrolysis like lysosomes, so sometimes that can happen as well. And there you can see that central vacuole in a plant cell. It can be huge and giant, and that will make the cell very turgid or full, rigid, or it can be really empty, and that makes the cell become flaccid. And those are terms that when we talk about osmosis, and when we talk about osmosis and selectively permeable membranes and such in lab, that's going to be a term that you're definitely going to need to know. So that endomembrane system is a complex and dynamic player in the cell's compartmental organization. And this is what it's made up of, right? Our nucleus, our smooth ER, our rough ER, our Golgi, our plasma membrane, our lysosomes, and our vacuoles. Mitochondria and chloroplasts are things that basically make energy for our cells. They are the powerhouses of the cell. They are the workhorses, they're often called. So mitochondria in animal cells, we go through something called cellular respiration, and that's where we take in oxygen and the oxygen makes energy. And energy for us is ATP. Chloroplasts, so we take in oxygen, we eat food, but we make this thing called ATP, which our cells use to do stuff. We think we eat in order to get energy done, but basically that food has to be broken down into something called ATP, and that's what our cells use. Chloroplasts are kind of like the plant version of mitochondria. They're in plants and algae. They're not found in animal cells at all, and they're the sites of photosynthesis. Plant cells do actually have mitochondria. And then there's another thing called a peroxisome. They're oxidative organelles. We'll get into those a little bit more. So mitochondria and chloroplasts display some similarities with bacteria. They are enveloped by a double membrane, so they have two parts of their membrane, just like that nuclear envelope did. They contain ribosomes, and they also have some DNA molecules within them. So when we said most of DNA is found in the nucleus, that's because some of it's in the mitochondria and the chloroplasts. And they also grow and reproduce somewhat independently within the cells. It's not really dependent on what the cell is taking in or what the other organelles happen to be doing. And so that leads us to this theory, the endosymbiont theory, which is that an early ancestor of eukaryotic cells engulfed a non-photosynthetic prokaryotic cell and formed this endosymbiotic relationship with its host. Instead of eating it, it just formed a relationship. So the host cell and the endosymbiont merged into a single organism, and that's how we believe kind of eukaryotic cells happened. It became a eukaryotic cell with a mitochondria. So at least one of these cells may have taken up a photosynthetic prokaryote, becoming the ancestors of cells that contain chloroplasts. So I would know, reread that write this down, I would know what the endosymbiont theory is. Mitochondria are in nearly all eukaryotic cells. They have a smooth outer membrane, and then their inner membrane is actually longer than their outer membrane, but the inner membrane is super duper folded to make more surface area. We call that the cristae. And we call the two membranes make a little space in between, and that's called the inner membrane space. And the very inside is the mitochondrial matrix. And the movement of electrons across the inner membrane space is what makes tons and tons and tons of ATP. So the cristae being present in a super large surface area makes the ATP, synthesizes the ATP. So you can see it here. We have our regular smooth outer membrane, kind of looks like a kidney bean. And on the inside, we have another membrane that's just folded, and that's the cristae. Chloroplasts capture light energy. So the chloroplasts contain a green pigment called chlorophyll, and they also have a lot of enzymes and some other molecules that function in photosynthesis. They're found in leaves and all the other green organs of the plant, and also in algae, and that's why algae is green. Chlorophyll is the color green, and that's what gives plants their green color. Chloroplast structures include thylakoids, which are sacs that are stacked. And so it's individual thylakoids, which are like big pancakes. And the pancakes all stacked together, just like at Denny's, you've got pancakes that make a grand slam, I think. The granum are the thylakoids stacked together. And then the chloroplast fluid or matrix is called the stroma. Chloroplast is one group of plant organelles called plastids. So here we see the thylakoids are each individual stacked thing, and they make up an entire, they make up the entire granum, and then that space in the center is called the stroma. Now, peroxisomes, that was the other thing that we mentioned really briefly earlier. These are specialized metabolic compartments. They're bound by a single membrane. So basically they use, this is what makes it so easy for me to remember. They use hydrogen peroxide, well, they're called peroxisomes, and they convert, they make hydrogen peroxide and then convert it into water. So when you leave your hydrogen peroxide out if you have ever noticed, hydrogen peroxide is always in a really dark container. If you were to have it in a clear container like rubbing alcohol, the interaction with the light would convert it actually into water. So that's why it's always in a dark container. And this is just a picture of a peroxisome. I think one of the last components of our cells that we're going to look at is the cytoskeleton. The cytoskeleton is a bunch of fibers that basically give strength to the cell, but also have some activities where they might move things around within the cell. You can see these things moving along these little strings. Those are vesicles and who knows what else moving along the cytoskeleton to get to other places. So it's kind of like a railroad or a zip line, something like that. So it supports the cell, helps it maintain its shape. It would be very similar to our muscles and our skeleton in our body. It also has some anchorage for a lot of the organelles and molecules. And it may interact with motor proteins, so it makes things mobile. Inside the cell, vesicles and other organelles can walk along the tracks, just like I showed you. And that is what we saw was the movement of it. This is what it looks like if you were to simplify it and blow it up. So I would know the three different types of fibers that make up the cytoskeleton. We've got microtubules, microfilaments, and then these little things called intermediate filaments. So microtubules are the thickest. Microfilaments are also called actin filaments. They're the thinnest. And then intermediate, guess what? It's things with somewhere in the middle range. There's a good table, if you want to go back and review this, that tell you the structure and function of the cytoskeleton with the microtubules, the microfilaments, and then our intermediate filaments. So microtubules are hollow. They're globular proteins. You can see in the picture, it's like a big PVC pipe. They're constructed from tubulin. Their functions are to shape and support the cell, and to guide movement of organelles. They're also really important in separating the chromosomes during cell division. When we get into that, we'll have a name for them, but they're actually called chromatin, and they're little microtubules. Well, we're going to talk about it, I guess, right now. So I would just know that these centrioles, I guess what I would know is that centrosomes and centrioles are microtubules, and all of the microtubules basically grow out from a centrosome, which is close to the nucleus. And centrosome is like the microtubule organizing center, and the centrosome has a pair of centrioles, each with 9 triplets of microtubules arranged in a ring. That part I don't really necessarily care so much that you know, but this is what it looks like here. Kind of an interesting thing is that we believe that these centrosomes and centrioles get weaker as we get older, and so we have a harder time replicating our cells properly. And so that's one reason why in older women having children, the risk of having a child with Down syndrome is greater than those who are younger, because it doesn't it doesn't split the DNA properly, and that's how you end up with the extra gene that results in Down syndrome.
Flagella, or singular flagellum, and cilia, or singular cilium, are microtubular structures that extend outside some cells and that assist in moving the cell or the cell's surroundings. Flagella are much longer than cilia. When a cell has cilia, their number is large, whereas a cell will have few flagella, or a single flagellum. Many protists have cilia, and the sperm of many plants and animals have flagella. Flagella and cilia are assembled from tubulin subunits organized to form a circular arrangement of 9 pairs of microtubules attached to a central pair. Flagella and cilia bend to cause movement of either the cell or the surroundings. The bending uses energy derived from the hydrolysis of ATP.
So that kind of gave us actually an introduction to this thing called cilia, which is a type of microtubule that things will move with. Flagella is another type of microtubule that moves, and so we know flagella because that's the tail on sperm. You can see them wiggling and moving there, and you saw the image before how it bends to be able to get it to move. This paramecium has cilia, and cilia are like itty bitty teeny tiny hairs that make it move around. So it's moving because those little hairs are bending. This is what the centrosome looks like. So cilia and flagella, microtubules control the beating of cilia and flagella. Flagella are limited to 1 or a few per cell. So they're like just a single or a double tail, but cilia are in giant numbers that cover the cell surfaces. And then their pattern is a little bit different in the way that they beat. But they do share a common structure. So a group of microtubules is sheathed by the plasma membrane, covered by the plasma membrane, and then a basal body anchors the cilium or flagellum, and then that motor protein called the dynein drives the bending movements of the cilia or the flagellum. And I think that video explains it so much better than looking at any of the pictures or reading any of these words. So microfilaments are thin, solid rods. We talked about microtubules. Now we're looking at microfilaments. These are the smallest, right? Thin, solid rods that are built up from these things called actin subunits, which is the substance that makes it up. The structural role of these guys: it bears tension, it resists pulling forces within the cell, so it is very similar to our skeleton. And bundles of microfilaments make up the core of microvilli in our intestinal cells. As an example, microfilaments that function in cellular motility interact with a motor protein called myosin. If we look back at our microfilaments, you can see here it's like sticky spider web or something like that. And so these things are pushed out. I guess the best way to think of it is it's kind of like the rods inside of a tent. While they're bendable and flexible, All together, they make the tent a pretty solid structure. And then intermediate filaments are larger than microfilaments, smaller than microtubules. They're intermediate, somewhere in the middle. And they're only found in the cells of some animals, so they're not found in any plant cells. They help support shape and fix organelles in place. And they're the more permanent of the cytoskeleton elements, and the other two classes are. So most cells synthesize and secrete materials that are outside, that go outside the plasma membrane. And so there's extracellular, outside cell materials that are involved in many cellular functions. And we'll talk about that as well as how these cells interact with one another also, because they've got to be able to talk to one another since they're stuck right next to each other. So the cell wall is an extracellular structure that distinguishes plant cells from animal cells, only found in plant cells. It's that rigid part of the cell wall that keeps it, maintains its shape. It also keeps it from taking up way too much water so it doesn't bust. Think about like when it rained and rained and rained and rained, and the ground is completely saturated and things are flooding. How come the plants don't just explode? Well, the cell wall prevents that. So it has multiple layers, or it may have multiple layers. You've got a primary, a middle, and a secondary. Every cell has a primary cell wall. It's pretty thin and flexible. It has a middle lamella, which is the thin layer between the primary walls of the adjacent cells. And then it might have a secondary cell wall, which is between the plasma membrane and the primary cell wall. Plasmodesmata are channels between adjacent plant cells. So if we look here, we still have a plasma membrane. Plasma membrane is always on the inside. The cell wall is always on the outside. Plasmodesmata are like little channels or little dashes or holes that can allow movement and talking between the cells. Animal cells lack cell walls but are covered by this extracellular matrix that's made up of proteins like collagen and fibronectin. They bind to receptor proteins in the plasma membrane called integrins. So there's this, it's like foliage on the outside of a cell, outside the cell wall, and you've got collagen, you've got fibronectin, you've got all of these components here that connect the two cells to one another and keep all of the cells kind of protected and in communication with each other. So I said that those cells have to have a way to communicate through each other, right? Either it's through direct contact, physical contact, or otherwise. So since they're touching, there's several ways that it's called an intercellular junction or a cell junction that allows them to communicate between one another. One is the plasmodesmata. That's what we saw here in the plant cell, just the channels. Then you have tight junctions, desmosomes, and gap junctions. So plasmodesmata are the channels, and what goes through it are like water and small things. Animal cells have three main types of cell junctions. They have tight junctions, desmosomes, and gap junctions. They're all pretty common in epithelial tissue, and that would make sense. Like it would need to talk to one another to tell it it's time to excrete oil or some other gland.
Desmosomes, or anchoring junctions, appear as thickened patches in the cell membrane region between two cells. Desmosomes contain specialized proteins such as keratin, the same protein found in fingernails and hair, that increase the rigidity of tissues. Desmosomes, such as those found in epithelium, smooth muscle, and many other animal tissues, are button-like junctions that bind cells together and also function as anchors for fibers in the cytoskeleton. In gap junctions, or communicating junctions, two cells are separated by a small gap, which is bridged by specialized channels that allow the passage of water and small molecules.
gap junctions help coordinate the activities of adjacent cells. For example, a hormone that stimulates one cell will often activate adjacent cells as well by the passage of intracellular signals through gap junctions.
Tight junctions, which bind cells together, are found predominantly in epithelial tissues, such as the lining of the intestines. Tight junctions form a barrier that prevents fluids from moving between the intestinal space and the space between cells.
Okay, so that's the end of chapter 4. All of these videos should be available under the Pearson website, and I think it's like additional resources or course information or something like that. You should be able to find it. Take course. I can't remember what it is, but it's the one that takes you to that other website, and there's a lot of videos and a lot of extra info on cells. Also, be ready to delve into this pretty in depth in lab.